My name is Michael Norris, and today I'm going to be talking about translation. So what actually is translation? Translation is the transferring of the information from a transcribed mRNA into an amino acid sequence. So whenever we do transcription, the DNA is transcribed into mRNA. mRNA is a sequence of nucleotides, and three nucleotides makes up one codon. And these codons from this mRNA transcribe an amino acid. This chart right here shows all of the possible amino acid codon relationships. These three here code for a stop codon, which ends translation, and this one right here codes for the start codon, which starts translation. So how does it work? It's carried out by tRNAs, which is transfer RNAs. Transfer RNAs are what actually add in the amino acid when it gets to the ribosome, but they also have something called an anticodon. This anticodon actually relates to the codon by having the opposite sequence, and so it can almost attach. So the actual process is we have our tRNA come in and attach to the spot on its codon where its anticodon matches. And so for this example, it's a start codon that codes for the amino acid methionine, and it's going to add in this amino acid and start the sequence. But now the second one's going to come in with its own amino acid, and it's going to attach with its anticodon. As this goes, it's going to slide over, if you will, and add in its amino acid into the sequence. Once this amino acid is added, it'll keep going over, this one would attach, and it'll keep going until we have a long chain of amino acids. Then a stop codon will come in and it'll stop translation. Then the ribosome will separate and we'll have an amino acid, and then the ribosome and the mRNA all separate. And that's translation.